Hello everyone, my name is Ashwini N, first year BSc CBZ student from JFGC Helanka. I am here going to tell about Human Genome Project HGP. Introduction to HGP. The Human Genome Project was an international scientific research project that aimed to determine the complete sequence of nucleotide base pairs that make up human DNA and all the genes it contains. It remains the world's largest collaborative biological project. The idea was picked up in 1984 by the US government when the planning started. The project was formally launched in 1990 and was declared complete in 2003. And the next is goals of HGP. To identify and map all the 20,000 to 25,000 genes in the human DNA from a physical and functional standpoint. To determine the sequences of the 3 billion chemical base pairs that make up the human DNA. To store these informations in databases. To discover more efficient technologies for data analysis. Allow the private sector access to the informations and technologies that arise from this project. Also to sequences the genomes of other organisms that are important in medical research such as mouse, Drosophila, etc. to address ethical, legal and social issues. And the next one is technical aspects in HGP. The process of determining the human genome first involves genome mapping or characterizing the chromosomes. This is called a genetic map. The next step is DNA sequencing or determining the order of DNA bases on a chromosome. These are physical maps. And the next one is sequencing strategies. To sequence DNA, it must be first be amplified or increased in quantity. Two types of DNA amplification are cloning and polymerase chain reactions. Now that the DNA has been amplified, sequencing can begin. Sequencing techniques used in HGP are: the first one is shotgun sequencing method and the second one is Sanger sequencing method. And the next one is mapping strategies. Genetic markers are invaluable for genome mapping. Markers are any inherited physical or molecular characteristics that are different among individuals of a population. A genetic map shows the relative locations of these specific markers on the chromosomes. An example of a marker includes restrictions, fragment length, polymorphism. Used in RALP markers or restrictions enzymes, these enzymes recognize short sequences of DNA and cut them at specific sites. Therefore, DNA can be cut into many different fragments. These fragments are the DNA pieces used in physical maps. RALP reflects sequence differences in DNA seeds which are cleaved by restrictions enzymes. And the last one is applications of HGP. The sequencing of the human genome holds benefits for many fields, from molecular medicine to human evolution, helps in identifying disease-causing gene, identification of mutation linked to different forms of cancer. The sequences of the DNA is stored in databases available to anyone on the internet. The US National Center of Biotechnology Information houses the gene sequences in a database known as GenBank, along with sequences of known and hypothetical gene and proteins, will allow for advances in agriculture through genetic modification to yield healthier, more disease-resistant crops, benefited the advancement of forensic science. Thank you for all.